Now, this led us into an area which was way over our heads. And again, back to the Facebook of science idea. For a small group, for a small laboratory, a focused laboratory to compete on a big problem like this, one must team up with other scientists. And we were very fortunate to team up with some very good groups, and I'll tell you about several. Shown here is Yoshinori Fujiyoshi and Andreas Engel. Yoshi from Kyoto University, an inventor of the world's best electron microscope, and Andreas Engel, a pioneer in membrane crystallography, who together with their outstanding graduate student, Tom Waltz, Tom is presently a faculty member at Harvard, solved the atomic structure of the water channel proteins. And in short, on the left is a panel showing the channel viewed from the top, a single aqueous pore, 3 angstroms in diameter, just big enough for water, water being the smallest biological molecule. And on the right is the channel viewed in cross-section. The top of the panel represents the outside of the cell, the bottom the inside. You can see a water molecule could permeate the bilayer freely through this pore. Here, a cross-section of the narrowest part of the pore, and this diagram was prepared by David Cousino in our laboratory. You can see a space-filling representation of water and the surrounding residues, the arginine above being a positively charged residue preventing the movement of protons, just large enough for water transport. Now, as a physician scientist, very interested in disease, physiology, and pathophysiology. And in a series of anatomical studies, we teamed up with Søren Nielsen, shown here from the University of Aarhus in Denmark, who's a superb microscopist specializing in kidney and kidney transport systems. It's a little faint, you perhaps can't see it, but on the top we see the immunohistochemical decoration with specific antibodies to our first protein, Aquaporin 1, which is in the proximal nephron, and the gold stain represents specific staining. What I'd like you to see, I'll try the pointer again. Pointers work well, but not that well. The brush border of the tubule is lined by this protein, allowing water to be reabsorbed from the primary urine back through the epithelium into the interstitium. And below is the second homologue, cloned by homology with our protein by a group in Tokyo. And this second homologue is very important because aquaporin 2 in the collecting duct is the vasopressin regulated water permeability. This is the protein that allows you to concentrate urine maximally when you're thirsted or release dilute urine when you're overhydrated. There are some significant clinical phenotypes which were identified because of the identification. Oh, here we have a pointer. Thank you. I think the problem is it's the white background. Yeah, either that I'm not a good at. Thank you. So in these studies, we teamed up with Landon King, an exceptional physician scientist at Johns Hopkins, who by some genetic analyses was able to identify individuals with knockout mutations in the aquaporin 1 molecule. And shown in the photograph here is a subject. I say subject because she's not a patient. She feels well, she looks well, but is homozygous for knockout mutations in the gene encoding aquaporin 1. She's a retired school teacher living in the south of France. I think the problem is the white background. Oh, a green one. That's a little bitter. So here's Landon, and here's our proband. Now, she looks well and feels well, but we were convinced there must be a phenotype because these proteins, if individuals are stressed, should uncover some weakness. And in fact, as you can see in the far right panel, normal individuals will concentrate their urine up to about 1,000 milliosmolar after overnight thirsting. These individuals can only concentrate it a little bit. So they can make it through the night, but will become dehydrated much sooner than other people. So it was an obviously important clinical phenotype. Now, several other members of the family were cloned. They formed 2 subsets, those permeated by water, shown above, those permeated by water and glycerol below. And I'll tell you briefly about a few of these. And each has an interesting distribution and often a predictable clinical phenotype. Aquaporin 0 expressed in lens is the basis for a number of congenital cataracts. Mutations, dominantly inherited cataracts, cause mutations due due to the opacity in the precipitation of proteins. And this is work of Masato Yasui, a postdoc in the lab who's back in Japan as chair of the pharmacology department at Keio University. Aquaporin 4 and brain work together with Ole Petter Ottersen from Oslo. The movement of spinal fluid through brain and the reabsorption is due to the aquaporin 4 protein. And in a very interesting set of studies organized by Mahmoud Amiri, we were able to establish in collaboration with a number of groups that in fact aquaporin 4 in brain leads to accelerated brain damage after ischemia. Now Mahmoud himself is quite an interesting character. He was a member of a university family in Iran that fled during the Islamic Revolution, and he grew up in a refugee camp in Pakistan. But being a very good student, he was adopted by the Norwegian Social Services, where he finished high school and became a medical student at the University of Oslo. And he went on to become a very distinguished, very successful neuroscientist in Oslo. And the studies that we undertook in the paper reported, as cited here, showed that the normal mouse and the mutant mouse having a defect in the bracketing protein alpha-syntrophin, created by Marv Adams Stan Freiner, when exposed to a defining brain injury, the mutant mice have more preservation of brain tissue, the rose color representing viable tissue, the pale representing infarct. And this has been reproduced independently by a number of groups. Now, brain edema is a huge clinical problem, often following a closed head injury or a stroke. And this work, and the work of other investigators, indicates that if we could inhibit or mislocalize the aquaporin 4, we might have an ability to prevent or ameliorate brain edema. Now, another member of the family, aquaporin 5, present in salivary glands, the apical membrane. So I'm pointing to the last membrane water crosses through this glandular structure during the secretion of tears, sweat, and saliva. And we think these are going to be involved in important human clinical phenotypes and may in fact be the explanation for the profound consequences during the great heat wave in Europe in 2003, affecting older individuals who have reduced urinary concentration, presumably due to defective aquaporin mechanisms. Now, the glycerol transporting homologs of the family have a virtually identical structure, and these structures have been worked out in great detail by a number of investigators, including Bing Yap at Berkeley and Bob Stroud and his team at UCSF. And in the pore lining residues, shown here, you can see the dark residues represent the pore lining amino acids in the water channel protein, and the pale residues represent the corresponding residues in the glycerol transporters. And notice, for example, this arginine is perfectly conserved of both. This is the size of the water pore. But in the glycerol transporter, this hydrophobic phenylalanine is not restricted. It has a smaller glycine and can open up, allowing the larger polyol glycerol to be passed through the pore. Now, glycerol transport is not something I'd ever really thought about, but it turns out to be quite important. And this protein, aquaglyceroporin 3, is important because it's in the basal levels of skin, where it is upregulated during wound healing and downregulated in skin, particularly sun-exposed skin, during aging.